Great. Well, it's a pleasure to be here and talk to you today about Haemato-Oncology applications of immune repertoire sequencing. And before I get into the content of my talk, I'd like to mention that immune repertoire sequencing data is a tremendously versatile data type, and it can be applied to immuno-oncology research, immunology, and infectious disease research, and as I will describe today, Haemato-Oncology research, where it can be used to detect and track B-cell and T-cell clones of interest. So for a bit more granularity on the presentation, I'm going to start with a background on immune repertoire sequencing before proceeding to describe some of the reasons why the field has migrated towards immune repertoire sequencing as a tool for Haemato-Oncology research. And then after that, I'll discuss spectra typing analysis as a tool to interpret repertoire data, before finally finishing the presentation with a discussion of somatic hypermutation and its impact on clonality and clone tracking applications. So when we talk about Haemato-Oncology disorders, those can be either of myeloid or lymphoid lineage cells. Within the lymphoid lineage cells, a subset are relevant to the immune repertoire analysis, and those are the T lymphocytes and B lymphocytes. B lymphocytes give rise to malignancies more often than T lymphocytes, and those are the primary focus of this presentation. Now, depending on the developmental stage of the B cell, one can observe a number of different cancer types, some of which are shown on the right side of this slide. B cells are typified by the B cell receptor. This is an incredibly diverse structure that is on the surface of B cells and can recognize antigen. B cells play an important role in neutralizing foreign invaders. The B cell receptor consists of a heavy chain and a light chain. The heavy chain, which will be the focus of this presentation, consists of a variable domain which is responsible for antigen recognition, but also a constant region which determines the effector function of the B cell receptor. Now, a B cell receptor is membrane bound, but the secreted form of the B cell receptor is called an antibody. And one of the reasons why there's such incredible B cell receptor diversity is through a genetic mechanism called VDJ recombination. And within this process, B cells actually alter their genome to create millions upon millions of different receptors. VDJ recombination refers to the recombination of tandemly arranged variable diversity and joining genes. This process imparts combinatorial diversity within the IGH receptor, but additional diversity comes from exonucleotide chewback at the ends of the V gene and J genes, but also the addition of random bases called N additions through the enzyme TDT. Now, apart from VDJ recombination, the sequences of the IGH can undergo further diversification through somatic hypermutation, which introduces point mutations to the IGH chain, but also isotype switching, which can swap out the constant portion of the receptor to one of 9 isotypes. And if you note the upper left-hand portion of this slide, a diagram of the rearranged IGH chain, you'll note there are FR or framework and CDR regions of the rearranged IGH chain. The CDR regions form loops that directly interact with antigen and play a primary role in determining the antigen specificity of the receptor, while the framework regions provide structural support. And it's been recognized by the field for quite some time that this IGH rearrangement is really an ideal target for tracking B cells of interest. And in this slide, I've highlighted some of the key developments in the analysis of IGH rearrangements. In the early days, the field utilized southern blot hybridization as a method for detection of B and T cell malignancies. That all changed with the advent of PCR. One observed singleplex PCR, where individual variable genes were amplified and then analyzed. But a big breakthrough came in 2003 with the development of the Biomed 2 multiplex PCR primer set. And this made the process of PCR-based analysis of the immune repertoire much easier. And you'll note that actually relatively recently has the field been applying next-generation sequencing to analyze receptor rearrangements. This was done initially with 5' race and Biomed 2 primers. And here it was demonstrated that this approach could achieve a limit of detection less than 1 in 10,000 cells. Now, the last point on this timeline, 2017, we introduced primer sets using AmpliSeq-based technology for immune repertoire analysis. And the key feature of this approach is that one can use many, many primers in a single reaction. So there's not the issue of primer-primer interactions that has historically made immune repertoire sequencing quite challenging. And so we are able to design primer sets that can amplify all of the known variable genes and alleles and thus provide comprehensive coverage of the immune repertoire. So with that, I'd like to introduce the Oncomine immune repertoire portfolio. We've developed assays for interrogating human T cell and B cell repertoires, but also tools to interrogate the mouse T and B cell repertoire. I won't be talking about those in this presentation. So we focus within the B cell receptor on the heavy chain because this is the most diverse in sequence content, and with the T cell receptor, the beta chain for the same reason. And within the human assays, you'll note that we have two types of assay, the LR or long read and SR or short read. LR refers to a long amplicon approach that provides a sequence of all 3 CDR domains. And this requires non-FFPE RNA input. The complement to this are the SR assays, and this targets the CBR3 region of the IGH chain or the TCR beta, and is compatible with both fresh and FFPE preserved material, both DNA and RNA. So with that, I'd like to, bring us to the second part of this presentation, which is to understand why the field has been migrating towards immune repertoire sequencing for Haemato-Oncology research. So I'll start by describing clonality testing and the related minimal residual disease research testing before finally proceeding to somatic hypermutation analysis. So clonality testing refers to a single sample analysis where one wishes to search for the presence of a dominating rearrangement. That dominating rearrangement is assumed to belong to a malignancy, and once identified, it can be used to track a clone over time. So this initial clonality testing through a diagnostic sample or pre-treatment sample is an essential part of the subsequent minimal residual disease analysis by immune repertoire sequencing. And this distinguishes immune repertoire sequencing-based minimal residual disease detection from traditional approaches like flow cytometry. So the reason why the field has been migrating to immune repertoire sequencing is that it allows one to achieve a very low limit of detection. 10 to the -6 has been demonstrated regularly, and this distinguishes it from other common approaches. So within the larger umbrella of rare clone detection, we have created the IGH-SR assay for rare clone detection and tracking. And you'll note from the cartoon on the left side of this slide, this consists of AmpliSeq primers that target the framework 3 region of the IGH chain and the joining gene region. So this provides an amplicon that spans that key CDR3 region, which is used to track B cells over time. The accompanying software that with this assay allows one to identify clones, but also clonal lineages. And these refer to sets of rearrangements that are believed to be related by descent, but differ owing to somatic hypermutation and class switch recombination. And through a number of studies, we and others have demonstrated that this assay is able to detect clones at 10 to the -6 limit of detection. So here I have an example of one such study. Here we took genomic DNA from a B cell line CA46 and spiked it into peripheral blood leukocyte genomic DNA at a frequency of 10 to the negative 6. 2 picograms of cell line DNA per 2 micrograms of genomic DNA. And from this large stock of material then, we prepared 30 libraries of 2 micrograms each and sequenced each using the IGH-SR assay. Now, 2 picograms of genomic DNA is approximately 1/3 cell equivalent. And so in an optimal situation, you might expect to detect the rearrangement at a frequency of 1 in every 3 libraries. And in fact, we detected in 10 out of the 30 libraries, very close to expectation. And from this data, you can back calculate the number of libraries needed to obtain 95% or 99% probability of detection at 10 to the negative 6. And this fits very closely with what others have observed in the field. So this really highlights the reason why the field has been gravitating towards immune repertoire sequencing for the rare clone detection application. Now, the other application I'd like to discuss is a somatic hypermutation analysis within the context of chronic lymphocytic leukemia. And it's been established that the level of somatic hypermutation of the variable gene can help predict the outcome of that cancer type. CLL having a V gene mutation of greater than 2%, sometimes referred to as MCLL, tends to have a better prognosis than CLL that has an unmutated variable gene or less than 2% somatic hypermutation. With that said, this is an imperfect prognostic biomarker, and there's a thought that potentially adding in isotype information of the B cell could further improve this stratification potential. And that follows from the observation that isotype switching involves the creation and repair of DNA double-strand breaks, and thus could serve as a window into the proficiency of the B cell to repair damage. So with this in mind, we created the IGH-LR assay for somatic hypermutation analysis. You'll note that, again, AmpliSeq primers targeting the framework 1 region of the IGH and the constant gene region. So this provides a readout of the CDR1, 2, and 3 regions of the receptor, but also the isotype. And this can actually distinguish all 9 isotypes from one another. So one can evaluate the somatic hypermutation through the variable gene portion of the amplicon, measure clonality and track clones through the CDR3, but then have that extra piece of isotype information. And I'll describe how that's can be useful. And as part of the development of this assay, we have compared the data from the somatic hypermutation data from this assay with orthologous approaches such as Sanger sequencing or NGS. And there's a very strong correlation as one might expect. Now, when I proceed into the next portion of this presentation, I want to introduce the concept of spectrotyping and how this may help interpret the repertoire data produced by the IGH-SR or IGH-LR assays. And the term spectrotyping has been around for quite some time. One of the first instances in the literature that you may find comes from a paper by Gorski in 1994. And this described a method for analyzing the T cell repertoire. And this involves a singleplex amplification of individual variable genes using FR3 and constant gene primer pairs. Each PCR product was loaded into a separate well of a gel and then visualized. And in healthy individuals, one would observe for each variable gene a roughly Gaussian distribution of band intensity, where each band corresponds to a different CDR3 nucleotide length. Now, in an individual with an abnormality, either autoimmune disease or otherwise, the plots would deviate from this Gaussian distribution. And on panel B of this slide, I have an example of myasthenia gravis. This is a T cell mediated autoimmune disorder where you're observing a non-Gaussian distribution. Now, I set this as context because with NGS data, we can take this type of analysis to a higher level. And here I'd like to introduce the spectra typing plots within the Ion Reporter companion analysis for these assays. So here you're looking at a spectra typing analysis from the IGH-LR assay. And in this plot, the rearrangements detected in a sample are displayed from left to right across the plot by the variable gene identity, and along the y-axis by the CDR3 nucleotide length. The size of the circle indicates the frequency of a particular V gene CDR3 nucleotide length combination, and the coloring indicates the level of somatic hypermutation. So you can note there's a lot of information in a single plot. And within Ion Reporter, these plots are interactive. So I can scroll my cursor over that plot and bring up extra information at any position. So for example, at the noted location, I've now brought up a pop-up window which indicates the V gene is V323, the CDR3 nucleotide length is 48 base pairs. The largest clone at this position has a frequency of about 1%, and with a V gene mutation of 13.7%. So what I'm showing you here is a healthy individual, a polyclonal repertoire, and you can note there are many circles in this plot that are about the same size. But now let's take a look at an individual with CLL. So here it's quite obvious the difference between the previous sample. One can observe a dominating clone that dwarfs the size of the other clones, the other parts of this plot. And you can note by the coloring that this clone likely has a level of somatic hypermutation greater than 0. And by hovering over it, we can bring up additional information. And here you'll note that at this position in the repertoire, the largest clone has a frequency of 44% and a V gene percentage mutation of 8.3%, noted at the bottom of the pop-up. So within CLL, this 8.3% V gene mutation would be interpreted as a good sign, and so a potentially better prognosis. Now you'll note at the top there are buttons that may be clicked by the user. In fact, with these plots, we can highlight, partition the repertoire by isotype, such that we're only viewing rearrangements having a particular isotype. So now, instead of viewing the entire repertoire, I'm going to click on IgM. And in doing so, I again see this dominating circle corresponding to the malignant clone, which suggests that the clone expresses IgM. Now I'm going to click on IgD. And here you see it again. So this CLL clone must express IgM and IgD, potentially as the product of alternative splicing. Now, the information that we just observed through the spectra typing analysis is also found within the clone subread tables of Ion Reporter. These are searchable, sortable tables, and each row of this table shows the features of a different clone. So you'll note that the first two rows of this table correspond to the two clones that we just observed, one expressing IgM and one expressing IgD. And these have the same level of mutation of 8.3%. And to the leftmost portion of this table, you can note a column that indicates the lineage ID. The software will automatically assign clones to clonal lineages based on the features of that rearrangement. So you can see that the first two clones are believed to be related by descent. And if we take a look at the lineage summary table, again within Ion Reporter, this provides a breakdown of the features of each clonal lineage. And so the lineage ID number 1, the most abundant clonal lineage, consists of two clones expressing IgM and IgD and having a combined frequency of nearly 80% of the repertoire. So clearly this is an abnormal sample consistent with CLL. So that was an easy case. Now let's take a look at a more challenging case. This was described to me as a case that was challenging to interpret through Sanger sequencing owing to polyclonal backgrounds. And you can observe why there's really not a dominating clone in the way that we saw with that previous sample. But here is where the isotype information can be useful, and this brings us to the important part of the second learning objective. With the isotype information, we can partition the repertoire, and that can help us hone in on an abnormally expanded clone. So at the top, I'm going to click on IgD. And when I do, you can note there's a dominating clone, while the other IgD-expressing B cells are quite rare. So this is abnormal, and it suggests that, in fact, the clone that's highlighted in this plot corresponds to the CLL, and thus one can more easily interpret the somatic hypermutation value of that clone. And just by contrast here, I'm going to click on IgA1 and look at those cells, and they are indeed polyclonal. So you can tell from this analysis that the malignant clone does not express IgA1. That looks normal. So now I'm going to switch into the third part of this talk, where I want to describe the impact of somatic hypermutation on clonality and clone tracking applications. And to highlight the importance of this phenomenon, we're going to look at a follicular lymphoma research sample. So to get back to what we discussed at the beginning of this presentation, B cells, unlike T cells, have additional diversification mechanisms after VDJ recombination, where they can undergo somatic hypermutation and class switch recombination. The somatic hypermutation introduces essentially point mutations within the VDJ region, which can give rise to any number of subclones. Now, in cases where a B cell malignancy of interest is undergoing constant somatic hypermutation, one could have a situation where there are many subclones that one may wish to detect and track by sequencing. So here's an example of a sample of suspected follicular lymphoma. And this, again, was a sample that was difficult to evaluate for clonality because, as you can note, there's not a clearly dominating clone. And this data was produced by the IGH-SR assay from FFPE DNA. But with follicular lymphoma, it's possible that the malignant clone could demonstrate ongoing somatic hypermutation, which could give rise to many subclones. In that case, we may observe an excess of clones having a particular variable gene CDR3 nucleotide length combination. And I can actually change the coloring of this plot to highlight the number of clones within the repertoire by clicking this button. And if this is the case, we may see an excess of clones then in one part of the repertoire. And doing so, with this type of analysis, you can really ask whether this sample truly is clonal or whether it's something more benign like follicular hyperplasia. So by clicking that button, you can note that there's something unusual here. One part of the repertoire has an excess of clones, as we suspected, and this may be consistent with follicular lymphoma. And the information displayed on this plot is also shown within the lineage summary table. And you can take a look at the first row of this table, lineage ID number 1. There are actually 38 clones within this clonal lineage, while the others have 1 or 2. So this is really an excess number of clones consistent with ongoing somatic hypermutation, creating this rich subclonal structure. And without a clonal lineage analysis, this type of feature would be missed because each of these subclones individually is at low frequency, but together they create the signal that helps one understand the nature of the sample. So from this, I want to convey that clonal lineage analysis is an essential part of interpreting the B cell repertoire. So with that, I'm going to finish the talk. To summarize, I described B cell repertoire sequencing and how it has emerged as an attractive tool for Haemato-Oncology research. This is owing to the great limit of detection, the ability to track clones, but also subclones. And then I also noted how it differs from traditional approaches like flow cytometry in that it requires a pretreatment or diagnostic sample. I discussed spectrotyping as a tool to interpret the repertoire, and further discussed how isotype information can help disambiguate certain CLL cases where one wishes to analyze the somatic hypermutation. And then I ended with a case study highlighting the need to identify clonal lineages within the B-cell repertoire data and how that can really help facilitate the interpretation of the repertoire. So with that, I'd like to thank you for your time, and really happy to take any further questions that you might have. Thank you.